Let's start. Maddie, are you able to give us update?
Hi, yes. So I was just kind of asking last night, and it seems like when I was at the lab, I guess a couple weeks ago, they said that they could teach me how to do the Epic after they got the Tempest data. And it seems like they have the Tempest data, but we're kind of unsure what to do with it. So I don't really know much about the data or like how to work it, and just kind of waiting on my part and how to do the Epic whenever they have a chance.
I see. Liam, can you give me context of?
Yeah, so we spoke with Hasang yesterday. So initially when Maddie came in like 2 weeks ago, we were still waiting for the Tempest data set for like Hasang to get access to it. So now she has the access. So we have access to the data through Hasang and Stas. And now we just wanted to figure out exactly which parts of the data set you wanted to kind of focus on for this particular project, because it's like the whole RNA sequencing data files are really big.
Right.
So it might be a little bit difficult, like technologically challenging for us to run the whole data set. So we were wondering if there were specific RNA sequences you wanted to like focus on that might help us kind of speed up the process and the data collection process. So I think Haesung can take over the explanation from here.
Yeah, I can just give you an update briefly. I think Stas will also join today later this evening, or like this meeting, to give you further update. But so he'll tell you about the OncoCyte. He's been contacting them. It seems like they are still trying to work it out for us. Try to see if the 27 genes match up with the Tempest data. And then I looked into the Tempest research, like the Tempest website, the section where they list all the publications, and I went through all of them to see if they have any like mRNA signatures that they've used to validate using their Tempest data. I only saw one relevant publication, and I showed it to Stas, and we kind of talked about it. Our plan is to see if we can divide our Tempest like patients into like responders versus non-responders, and try to see like if there are any like prominent genes that can predict immunotherapy response. But I think he will give you further update on that, because I don't fully understand like the technical stuff that goes into it, like how to further like really analyze the mRNA data and stuff like that. And then it seems like, and then Liam also told me yesterday of how like Yuri's AI model can be used to analyze the mRNA data, and it seems like your plan is to use the Tempest data to validate that one. And so it seems like we're using the Tempest mRNA data to validate 3 different models, like OncoCyte, and then the publication that I found on Tempest, that one, and then Yuri's AI model.
So Maddie, when I first mentioned the project, did I mention OncoCyte?
Yes.
Yeah. So this is where we are. I think OncoCyte has their own signature, which just uses how many genes?
27.
Yeah. So that was going to be pretty easy, like just extract 27 gene expression level and send to OncoCyte, and it's patent protected, right? So they will give us their prediction based on 27 genes, and we're going to test whether that prediction is true in our cohort. So it was going to be very simple, but they're saying Tempest data does not have that 27 gene in total. Like some genes are lacking. So they're saying they can't run it, but they'll figure out a way to do something about it. So that's where we are with Oncocyte. But I remain hopeful that something will come out of it. So that one is in limbo. Number two, Tempus, they have their own algorithm to say this is like immune, like hot tumor, cold tumor, just based on mRNA. And Haesung, do you have that article with you? Can you pull it up so that we can see it?
1 second.
So the second is use what's publicly available. Not patent protected, but anything that has been explored before. And the third is let's make our own prediction model using AI. That's what I told Dr. Jisang yesterday. You can share screen.
Sorry, it's been a while since I read this paper. I can get back to you on this.
Oh, it's not this one, but you have to
No,
find.
I can get back to you on this, yeah.
Okay.
Yeah.
So as you can see from the similar articles, you can find mRNA-based signature for non-small cell lung cancer. There's few articles that are relevant. And if they're funded by NIH, they should be able to share their code or how they analyzed, how they came up with those signature. But easiest thing is, like you said, if Tempus is already doing it. So Maddie, do you have access to our Tempus portal?
No.
So Liam, why don't you give it to her? I have my ID for that. And if you go there, there's a FDA approved report, but also there's a research section where you go in and it talks about HLA, immune cell infiltration, all that listed there. And then there they list immune cold tumor, hot tumor. Why don't we pull it up? Haesung or Liam, can you go into the portal and share your screen so that we can look at it together? But the point is, whatever Tempus already used to classify patients based on their mRNA immune profiling, we'll use that because we can tell Tempus to just send out among these patients, just give us a Excel spreadsheet that has the outcome. And we can use that to correlate with patient data. So Maddie, your role is number one, because we'll work with the company to get all these data.
Okay.
Your role is to make sure clinical data is clean, meaning response, progression-free survival, overall survival, what were they treated with agents. All these data has to be collected, and a lot of them are already collected. But I think what's been collected has been, collected maybe a year ago. So we might have 1 year uh patients to collect. There's things like that. Okay, so here,
You can
maybe,
export data item.
yeah, maybe go.
I'm looking at
to-
only the.
That's Sarah. That's yeah. Click this one.
I just filtered out only the patients with the RNA data.
Yeah, so you go down. Here is immune biomarker, TMB microsatellite, additional information, treatment implication, potentially actionable variant.
Oh, the RNA report is not available yet for this patient.
Oh,
Oh, wait. Oh, okay, yeah, so this
oh,
one.
so this is the, yeah, RNA
Yeah.
expression. So what we're trying to get is just as overexpression. We're trying to get actual standardized or normalized expression level for each gene, the raw data of all the genes, and then go click on the immunotherapy. So it says research use only. This is what we want. MUTYH frame shift. So this is, I think, is going to be important for mucinous lung cancer project. This is the mucin gene, and I do believe it's important. Resistant risk. Oh, what happened?
Oh, sorry.
Certain gene alteration have been associated. So this is like, so click here.
This
Oh,
one?
no, not there. Resistant risk. The one with the blue
Oh,
color. Click here.
okay.
So these genes, interferon alpha, beta,
Can't go down.
go down, and
I can't go down. I don't know why.
oh. Anyway, these are the genes that are published to be linked with DNA mutations that are linked with immunotherapy resistance. So you could come up with whether there are mutation listed in, present on these genes or not. So this is a really good list for the research. The TMB is here, this HLA. So whether HLA is homozygous or heterozygous has impact on whether immunotherapy works or not. This was Dr. Ho-Ryun Choi's initial work, and he published that article. But this is important too. Go down. Here are the immunotherapy targets. And this is just based on RNA expression level. What percentile? RNA compared to tumor tissue. So go down. So there are a lot of different immune checkpoints. So these levels are important. Go down. This is immune infiltration. This is like X cell or the total immune infiltration. And then there's each cells. So this is the exact same thing we did with our TCGA paper. This is just based on mRNA, not IHC.
Mhm.
Go down.
That's it.
Oh, that's it?
Yeah.
Oh. They used to have immune hot tumor or cold tumor or something like that, but I guess they don't now here. Oh. Oh, click on RNA. What is an RNA?
Not available. What's the difference between RNA and RNA expression?
I'm not sure. Can you go to other patient and see what it shows?
Yeah. Mm,
Well, this is liquid.
oh, this patient?
addendum, RNA sequencing, no gene codon, no gene arrange. Oh, so I think this is a fusion report.
Mm.
RNA sequencing can lead to better identification of fusions. And expression is expression. So this is really a gold mine. We have a lot of breast cancer, bladder cancer, but we just have to ask the right question. Click on immunotherapy. See if this is same. So PD-L1 expression, mismatch, repair, resistance, TMB, PD-L1.
Yeah.
So total immune cell composition is 21%. So you can argue this might serve as something like Lunit score, like immune cell infiltration level, how hot the tumor is. And you can use this as a continuous variable to see if this predicts immunotherapy response, whether the total composition or the T cell. So any of the values you can use to see if that helps us to differentiate durable responders versus non-responder. And the way to differentiate those, Dr. Jisang can help because we did the exact same thing with radiomics. How do you group them into two different groups? So you can do what exactly Dr. Jisang have tried. So the question is same. He used radiomics as a biomarker, but here we're using mRNA as our biomarker. So any signature that comes out from mRNA, whether it's this immune cell composition, whether it's a different signature like oncocyte signature or anything different.
Yeah, so this
Here-
was the paper that I was showing you earlier.
Okay.
So they used the RNA-seq data and then matched clinical data, create a signature that and see if there's any association with immunotherapy response.
Mhm.
And then they also evaluated the CD8, CD4 T cell infiltration, and then saw whether their CAN T cells had a toxicity signature could be used to predict response. Developed a gene signature consisting of genes that were highly expressed by CD4, CD8 T cells. The gene list includes 25 genes. And then, so these are the graphs that generating using the cellular plastic gene signature. This was the paper that I showed that.
So I guess they extracted those genes. So whatever they found is the cytotoxic score was not correlated to Excel estimate of T cell infiltration.
Mhm.
Mm. Okay, go down. So here there's ICB treated. So is this cohort publicly available cohort, or is this something that they have?
I think it's their Tempest people.
Oh, this is a Tempest cohort. Okay.
Mhm.
Do you know how many of this?
documentation? Oh, wait, no.
Real world 123 non-squamous were molecularly profiled. So they did not only non-squamous 123, right?
Mhm.
So that's very interesting. Mm. That single cell was done on 10. It's so expensive. Okay, I think this is a nice paper for us to understand what Tempest did. There's more than mRNA here. There's a lot of single cell, everything.
Mhm.
When was this published?
2022.
Oh, so pretty recent. And this is Nature Communication. Ali Khan. See what you see in Tempest?
Mhm.
Tempest, okay.
Chicago.
Okay, so it's really the Tempest paper. Do you know Amin? I know him. So Amin works for Tempest, but he's also a faculty at UIC, where William
Mm.
Pei is doing Hemong Fellowship.
Mm.
So he interviewed William.
Mm.
And yeah. So it's amazing how he could work for a company and also work for a university at the same time.
Mm.
But anyway, he did his MD, PhD, and Hemong Fellowship at Stanford.
Mm.
So he does a lot of translational work. Anyway, maybe Haesung, you can do a journal club maybe in next couple of weeks. Just read about it and understand what they've done. That's all you need to do. But I guess here they have an algorithm they already made. So they looked at different, CD8, TD8, they all cytolytic score, everything, and whatever correlated the best. You use that algorithm, and you can email Tempest, hey, for these patients, can you send us the score or whatever your prediction model's outcome? You can run those. We just want to validate whether that is working in our own hospital data. So we can do Tempest algorithm based on this. They've already published this. It's not a secret. But whatever it is, it's for their benefit. We're going to promote their signature if it works. If it's not working, then it's a different story. But I'm sure it's going to work. So that's the second project. And I think Maddie can be involved in either one, but it takes time for us to get the signature. But in the meantime, we need clean clinical data. Once the signature is out, Maddie, you should be able to correlate that clinically, who's
Okay.
responding. Yeah.
I don't really know how to do that, like in terms of logistics.
Right. So you'll come to lab one day and learn how to do it.
Okay.
Yeah. And Liam, you'll be able to, you and whoever's in the lab at that day can work with Maddie to train her for clinical data acquisition. But this is like mRNA based project. Like I said, it has 3 different approach: 1 oncocyte signature, 2 Tempest signature, or any publicly validated signature. And I gave you an example of Tide.
That's tied. Yeah,
Yeah.
we're also trying to work that out as well.
Yeah, and that's publicly available. It's a signature developed by
Mhm.
Dana-Farber Group. So we can, so our role is, what signature works best in our cohort. And then once we're comfortable, we found something that's interesting and can be validated. That in itself is a validation paper, but can we go beyond and develop our own model? And the way they make model is not through AI. So let me tell you, this is a lecture for you guys. There's a top-down approach, bottom-up approach. And okay, the other way to put it is there's a hypothesis-driven approach. So you think that CD4 and CD8 are most important cell, just based on what you learn. And you only look at that cell infiltration and try to correlate that with response. That's great hypothesis, but that could be your greatest bias too. There may be unknown genes that would play a biggest role and you just don't know it. And maybe it's the interaction, like this gene is not important when this gene is not expressed. Only when it's expressed, this gene plays a role. There's so many interactions that we don't know. We have no clue. And we can't come up with any biologically plausible hypothesis every time, when we get new data. So we let AI figure that out. And AI is perfect. And not only in imaging analysis, but also in figuring out raw biological data. Any data coming out from living organism. It could be heart rate. It has already figured out in ICU, if heart rate changes this pattern, the patient's likely going to die within 30 days, just by analyzing the heart rate changes. And human eyes cannot figure that out. Okay, that's where AI comes in. So what I'm suggesting is mRNA data as like everything, every gene and every expression level. Don't try to trim things out by your own hypothesis or bias. But you rather use every value that you get, the raw data. You can normalize it. You can run it with normalization. You can run it without normalization. See what happens. But the raw data, can we use that to predict response? So that's where AI can come in handy. And Dr. Jisang Yu is building his expertise in this field with his radio experience. So work with him. The greatest benefit of working with Tempus is we always have access to raw data. So with the raw data, can we develop AI model that predicts response to immunotherapy? And not only immunotherapy, the immune toxicity. Can we develop model for immune toxicity? So that's the third approach. But Maddie, whichever model is used, we need clinical data, clean clinical data. So you can work on that while we work on, finalizing these results.
Okay.
yeah.
That sounds good.
So I think Liam is still your contact, but Haesung will work with her with you because yeah, it's also part of her work scope too. So Liam and Haesung will be your mentors on this project.
Okay.
So yeah, choose a date, come in, they'll sit down with you and go through how to get all that Tempus data up till now, because I think we've gathered a few more patients.
Okay, sounds good. Thanks.
Great.
Dr. Che, just a question. So when we're collecting the clinical data, do you want us to just collect everything that's on the Tempus website, or do you want us to focus on like the genes that were, reported to be associated with resistance? For example.
Yeah, I think everything in the immunotherapy section
Mhm.
we need to collect, and it might be easy to ask Tempest to extract that data for the, let's say, the first step is come up with the full list of patients who meet the criteria. And then ask Tempest to extract all the immunotherapy related information. And then it's the same thing, response, PFS, OS. And a lot of it will overlap with radiomics.
Yeah.
So build on what we have to come up with that list. Yeah, and information.
Are we also gonna get the toxicity data?
What
Whether they
data?
developed any adverse events, so we are adding toxicity clinical data.
Yes, yes. Once we're collecting it, we should collect the toxicity data.
Gymnastics.
But it's going to be difficult because this toxicity, in my mind, has nothing to do with tumor RNA. It's more to do with normal DNA. So I don't know. Oh, Stas, you're here. You know how DNA sequencing world, we have tumor sequencing and normal sequencing, and then comparison will give us an idea, like the MSI high is, extracted that way.
Yeah.
mRNA, is there a way to sort of extract both tumor and normal tissue expression, nearby normal expression?
I mean, in the past, so the way it's been done at some of the companies I worked at is the team will get 2 samples. They'll get the
Oh.
tumor and then they'll get adjacent normal. And then they treat that adjacent normal as healthy cancer-free tissue, and that functions as that base negative control. But not all what Tempus requests from the clinicians and from the surgeon. I'm gonna take a look at the Tempus, although this isn't directly helpful, but they do have germline for
Right. Yeah, maybe ask Tempest about how they can help us get the normal tissue expression. I know they have DNA from normal, so I feel like normal tissue, the RNA have more information to predict toxicity. Yeah, maybe. So Stas, can you ask that question? Maybe CC me about like a normal tissue RNA sequencing, whether they have ever done it or.
Yeah, absolutely. I just reached out to them about a different question, and it sounds like the person who was away for a little while, so she said she's gonna get back to me about my first question, and then I can follow up with this question. I kind of don't want to overload her.
Yeah. Just ask.
But yeah, I'll make a note.
Okay. So Stas, any insight into what I just said about like using AI to analyze mRNA and further DNA? I think the key is we need more patient.
Right.
So, I think Tempus project, like there's a Tempus project that will allow you to look at maybe like 500 patients at max at given point. Their paper was published with 123 patients.
Yeah.
So if we can, get up to 500 patients, that would be really good. But even, I think now we've gathered enough data. And then the other thing is, I know signature may be unique to non-small cell lung cancer, but bladder cancer, respond very well. Triple negative
Mhm.
breast cancer respond very well. So, at some point we can look at other immunogenic tumors and look
Right.
at signature together. That will because that's Lunitscope is also pan-tumor biomarker.
Yeah.
So we could come up with like a pan-tumor immune biomarker. That way we could easily get up to 500 patients. But let's see, until now, let's say how many patients meet this criteria for immunotherapy. And it's tough because we're not doing immunotherapy only for lung cancer these days. It's usually combined with chemo.
Right.
So that signature might be very different from immunotherapy only signature. So there's a lot of challenges there, but any immunotherapy containing regimen will be collected. And for to make data clean, we could start with immunotherapy given as a first line. So it's very similar to the radiomics. So built upon that database.
There is actually one paper I wanted to mention to you that Hesung found last
Uh-huh.
week that actually kind of does that. They have a pan-cancer cytotoxic T cell signature.
But is it pan cancer or just non-small cell?
Well, it was initially before they tested it in non-small cell, it was pan cancer, or sorry, excuse me, pan T cell. Yeah, it was a pan T cell cytotoxic signature across all T cell types, but it looks like they only tested it in non-small cell lung cancer.
I see.
So, and it was based on the Tempest.
Right.
Tempest.
I told some to do a journal club.
Okay.
With it at some point. We just skimmed through it, but
Okay.
yeah, Hesung work with stats to get that. Once we have a full list of patient we want, email Tempest to ask for that signature.
Well,
That-
it looks like based on this paper, the signature is just they talk about how they do it. They have a list of 25 genes, and then they calculate a cytotoxic score based on the expression of those genes.
Right. So
and it's just the mean.
Right. I mean, Stasi, you can extract it and just run it as they said.
Yeah, I can definitely
Yeah.
try.
But we can also have them extract it
Right.
for us so that we can compare, make sure it's like a double check. That we did what they exactly did. So yeah, that's an easy first step. See, that signature correlates with our outcome. So Hesung Stas and Maddie is our medical student who's gonna help with this project. But I'm excited for our third approach, which so Jisang, you could sort of play with there's like mRNA data from TCGA, and there's like some you can play with let's say extract 20 patients from Tempest. And in 50 you can extract, download it. It's not a lot of data. Even 100 patients you can download. What I want you to do is just play with the data, extract it. You can do it from your laptop, and then that as a raw variable, and then you can randomly assign responder, and you can kind of see how it works. But I would suggest start with your own radiomics data, because a lot of them will have Tempest. So get the Tempest available patients list, and just download or RNA only data. But it's not RNA only, because I think there's going to be, like I said, TMB PDL1 is already identified do the PD-1 as well, not just PDL1. So when you, and then also when you do PDL1, you can request immunohistochemistry and RNA expression both. But PD-1, you will only have RNA expression. And TMB, all that data can be utilized to run AI modeling. And you already know the clinical outcome for your radiomics patients. So Jisang, I think you can start there. Just get the raw RNA-seq data, work with Stas to get it. You have the patient identifiers for all your radiomics patient. So use that patient first, get the Tempus data, and just run the same analysis you've done. That's easy start, and just play with it. See what you can find. The old-fashioned way is what Myeongwoo did with intervent data, which is, you know, responder, non-responder, high expression, low expression, where's the biggest delta, find the biggest smallest p-value, and find the gene set that, you know, has the p-value cutoff of point something something something, and then use that set of genes to create heat map and then separate. So you can do that too. You can do that with Myeongwoo, Jisang. Okay. But that's the old-fashioned way. But I don't know if AI can do a better job than that. So I think we have to build confidence. What's the best way to analyze? But I'm sure with Tempus, you know, if we get more data, if we get to like 1,000, you know, I think we'll get much better model. It's just the way. I think, but with more than 100, the model should be able to show something. Even good old-fashioned PDL1 TMB separates the curve very nicely. So with more parameters, we should be able to see something. So let's work on that, Jisang, and Myeongwoo can help as well. Okay, let's go to Stas, updates from you.
Yeah, so other than that, the only other update I had as far as the projects go is regarding Oncocyte. So I've been chatting with them about how we can make this work, because it wasn't clear if Tempus was actually sequencing the genes that they were part of their signature. And they said that as long as those genes are covered within the transcriptome that Tempus sequences, or at least gives us data for, they'll be able to do it. I went through and I manually looked to see if those genes were present in one of the patients from 2021, and none of the genes were there. That could just be unique to that patient, or that could be that just how their transcriptome analysis goes. So I emailed them and I said, hey, is there any way you can, in an automated way, look through all your patients and say which patients express these genes? And that's what they told me they'd get back to me about. If the answer is yes, and they can give us a list of patients that have gene expression of those 27 genes, then we can do Oncocyte. But that's really what it all depends on.
I see. But that 27 gene is in the list of the genes they ran, or?
So with transcriptomes, I don't know how they do it on their end, but generally speaking, what you will do is you will either pull down all the RNA using poly-T, and then to pull down poly-A, the tail on mRNAs, or you get rid of all the DNA using DNase enzymes, and then you sequence what's left. So the question is, in that batch of RNA that they're concentrating, that they're pulling out, are the transcripts for those genes being captured? I don't know. And so that's the real question. If they are being captured and sequenced, the next question is, are they being sequenced to enough depth to be able to say that they're there or not?
I
And
see.
if that answer is yes, then they're there, then we're measuring them.
Okay, okay.
A little bit technical, but the point is that they should be able to tell us.
Yeah, okay, so that is that, but we still have a lot of backup options that we could play with, so, okay, great.
I'm still looking into Tide. It looks like at least it just seems to be very focused on this notion of adjacent normal and tumor tissue, which is a great approach.
Right.
Unfortunately, TCGA doesn't offer both a normal sample, at least for all the samples, all the patients, a normal sample and a tumor sample. Tempus might allow for that. Unfortunately, when I look into the RNA folders, there's only one sample. There's only one file, RNA
Yeah.
file for every sample.
Yeah,
So that's something else I need to ask them about to see if maybe they already do that normalization. And basically, I just need to do a little bit more investigating to be able to rule out Tide for certain.
okay. So, yeah, keep asking, but at least the Tempus signature you can do, maybe, yeah, that's low hanging fruit based on that paper.
Oh yeah, and then that paper has that list of genes, and we can. The thought was to have patients who are responders, non-small cell lung patients who are responders, who aren't responders, and then just look at the expression of those genes between those two groups.
Right, yeah. So, yeah, maybe do that. That's a very low hanging fruit.
Right.
And with Tempus data, we know it's feasible, so you do that. But let's say, what we're trying to get to is multi omics. So mRNA, maybe that. You have TMB, you have PDL1 from protein, IHC.
Yeah.
So, prediction, one layer,
NPH.
adding layer, would that increase your precision
Right.
model? So those are all easy things you can kind of play with.
But they rely on us having a non-small cell lung cancer data set that has responders and non-responders.
So their Tempus data is not reliable, do they? Because they just go by text mining. They don't require investigators to fill out, they're not accurate. But I guess overall survival, it has to be accurate because you either alive or dead, right? So at least overall survival data, if Tempus is willing to share with us, that's great. So I think the best scenario is, use our own data to validate, but also use Tempus data because we don't want to just validate and stop. We want to add layers of multi-omics and then use whatever Tempus cohort that they can provide as a validation cohort to, at least do overall survival prediction analysis. So let's do that as like a bigger picture. But you heard what I said about AI. And Stas, you're welcome to be involved as much as you want to be on this. And Dr. Jisang is leaving soon, but he's gonna teach us how he ran some of the radiomics analysis as well as the mRNA AI thing he's gonna run. So you're more than welcome to connect with him to run the codes. And you run it yourself. This would be a powerful tool.
Yeah, it's something I'd really like to learn. Definitely a gap in my knowledge at this point on how to set up and train those models.
Yeah. So yeah, we're gonna just have more and more data, and it's going to be and then we're gonna have huge data. Oh, so the other thing is this: like all these models, papers. Let's say Tempus. There's a ICB cohort, immune checkpoint blockade cohort, and this paper or many other papers will use publicly available clinical data. That's what Swanton did. So continue to look for those papers that will give you mRNA sequencing or DNA sequencing and all clinical variables, PFS, OS.
Right.
You'll be amazed. And they're all lung cancer patients treated with immunotherapy. And those Excel spreadsheet is online, like you can download.
Oh.
And that's how a lot of these statistical papers are generated. And they say validation code A, B, C, D, and they're all previously published in Science, Nature, you know, in that caliber. So just read those papers to understand where you can download those data set. It's available for sure.
Mhm.
And use those cohort for your research. It's a validation cohort. So
And so you're saying the data is accessible, the DNA-seq, RNA-seq,
yeah,
or you're talking, okay, interesting, okay.
yeah, you just have to find those cohort. It's all in NIH funded research.
Okay.
It's requirement. You can't publish.
No, I believe you. I'm just
Yeah.
surprised. Yeah.
Yeah. So some good papers are just coming out from pure bioinformatics lab, and they've
Yeah.
never ran trials or treated patients with immunotherapy, but they have access to these data and they just
Right.
pull it and run it this way, that way, and they still publish in Science. So yeah, so that's the other piece. Anything else?
The next thing is, earlier this week, Liam sent me a list of patients for Liesel's project.
Mhm.
I've sent back some PyClone output for about 16 of them. I'm gonna keep trying to see if I can generate output for the rest. Some of them just don't have any Tempest data. And
So
then
this is
some,
all Tempest sequencing DNA?
correct.
Okay.
And so there's 16 that I've sent them data for, or sorry, output, PyClone output for.
Mhm.
And then I'll try and do the rest or see if I can get the rest working.
How many are you gonna do in total?
Well, the list they sent me has, and then Liam, feel free to chime in if anything has changed or you have any updates. It looks like there's 61 in this
Okay.
list. And so, yeah, I'll try and get as many of those or PyClone for as many of those generated as I can.
Okay, great. So if I remember correctly, using different cutoffs, you'll have different clone numbers.
Well, the way the output looks is I can show it, but in a nutshell, it gives you a list of clusters. Each cluster has a number of mutations associated with it. And so the way that I've been doing it is if a cluster has at least 2 mutations, I treat it as a real cluster, and then I count those real clusters. There's a lot of other ways to do it. Other groups have come up with ways to define what a clonal cluster looks like, what a subclonal cluster looks like, and then use that as another dimension to differentiate patients. Some look at specific mutations within clusters. So there's a few ways to go about it, and I think we can get creative with it. We just need to, decide on something.
Okay. Yeah.
Was that your question? Sorry if I misunderstood.
I was just hinting, the way you define clusters will bring out different, outcomes. So
Right.
If you can sort of generate, a few scenarios, this definition will spit out, this clusters.
Yeah.
And so that we can do, different analysis using different criteria or something.
Right. Well, at the very least, or at first, I can look into this distinction between clonal and subclonal clusters or mutations or something like that. And then, because the raw data is the same, regardless of what what cluster filters we use, what mutation count, everything is the same. So I don't have to rerun it. The question is, what are some things we want to try?
Right. So how many, like, for one patient, like how long does it take for your computer to?
Well, I do it on the computing cluster, the Northwestern computing cluster. And so it really depends on the patients. If the patient has 5 mutations, it'll take a few minutes. If the patient has hundreds of mutations, it can take 12 hours. And the nice thing is I can run all the patients in parallel.
Oh,
But it still takes as long as the longest patient. And the way I've been doing it, I don't know if it's correct or not, but this output is variable because it's a little bit random. It
yeah.
starts in a random place and it starts clustering. So the output can, for example, I was looking at the output that I sent Liam and Liesel, and there are patients who are sometimes set to have 1 cluster, and other times it's 3. And when I look to see the kind of the distribution, there will still be 1 giant cluster, but then there might be 2 small ones that have 2 or 3 mutations. Do we count those? We decide. But the point is that I've been running each sample 3 times on 3 separate days to try and kind of distribute the randomness and or try and control for the randomness a little bit. So at this point, it takes me 3 days to run it, assuming that no sample takes longer than a day to run.
Got it. So Haesung, once you get your NU ID and have access to the cloud and everything, meet up with Stas one day and learn everything he's doing so that Haesung can kind of run it.
Right, absolutely.
Yeah.
The other thing
I'm still
that,
waiting for it.
yeah, go ahead. Sorry.
Go ahead.
I was
No,
gonna say
Haesung's still waiting for that.
the other part of running PyClone, the other half that can take some time is setting up the input. So this is where a little bit of programming experience can be really helpful because you could just automatically create these input files. You can do them manually too. It just will take a little longer, but that's another kind of half of it that you'll need to learn.
If there's any like useful YouTube video or any like material that Haesung needs to read before meeting you, that to expedite learning experience, then yeah, please do so. About that programming part.
Yeah, I can send you the link to so they have a GitHub page where they kind of host all of their software and have a little like README that talks about what the files look like, what information goes into them, how it's organized. My plan was to go through that with you when we meet. And kind of just show you what the files need to look like, and you can decide if you want to do it manually or you want to name your favorite programming language and do that, and I can help you. And then once you have access to the cluster, I can then show you how to run things on there. I can even give you some of the scripts I'm using because they're very generic, and then you can go from there.
Sounds good. Okay, and
yeah, then what the last update is regarding the TMB ITH paper. I've written the discussion and just wanted your feedback on it.
Okay. Just,
and I can send the link in the chat.
Yeah. Okay, I will share screen. Okay.
And then if you just click on the discussion, it'll take you right there.
Okay. So which part is this?
This one, yeah.
The 3 paragraphs?
Yeah.
Okay.
I'm not sure how long it should be, but I tried to write more so I can cut
Sure, so ITH exhibit differential prognostic value in histological subtypes of. That is true. Fine. Hmm. Hmm. So is this true? What about Swanton's paper? He had clonality, index in TCGA, if you remember his paper.
Right, let me confirm, but it seems like every paper I've seen out there, at least every pan cancer paper I've seen out there that uses TCGA data,
Mhm.
doesn't show a relationship. It's neutral. There isn't a positive or negative relationship.
Hmm.
Like the papers that I cited. Let me just look his up. Charles Swanton.
Or you, because I'm pretty sure Charles Swanton's paper looked at non-small cell. Maybe you can phrase it, express it in a way that
Yeah,
they looked at non-small cell lung cancer as a aggregate,
right.
but no prior work has, delved into this question of, what is the clinical implication of ITH among different like squamous and non-squamous.
Yeah,
I don't think, right?
right. It's never been, well, the only paper that did do that, I think it's number 34 or not, maybe it's 9. I forget which of these two. They did a whole pan cancer analysis across all TCGA, and
Hmm.
they did divide it up into LOD and LOSC, and it didn't show any differences.
This
It
one?
might not be that one. It might be number 9, but regardless, I can pull it up. But you're right. I'll emphasize that more. Yeah, I think that
This
one
did
is,
not pan out to be a good predictor. I think, is this TCGA data?
yeah.
And wasn't a good predictor?
It was in head and neck.
Uh-huh.
If you take into account HPV,
Oh,
I think it was also a good one in melanoma. I
okay.
forget, but there were some indications where it was a good predictor. Just not in
This one,
squamous.
was this TCGA as well?
I do not remember, actually, to be honest. Let me
Let's
confirm.
pull up this one, because this is more recent.
Okay. Let me just look. Yeah, this was TCGA.
Yeah, why don't you share screen?
Yep.
Of the. Let's just go over the
Yeah.
figures.
All right. So yeah, 9800 TCGA. 14 tumors showed that high ITH associated with shorter survival. Confounding factors, these weren't confounding factors. And then they also did a meta-analysis here.
Show me. Is that meta-analysis using different papers?
Yeah. So
BRCA, that's breast cancer.
and here we have LOD and LOSC. And as you could see, the confidence interval crosses 1 in all of these, so they're not significant.
But what's the combined one? Go down. So if you add them all together,
Right.
it's like 1.65. Okay. So guys, this is what I'm talking about. These are, so you have to calculate ITH, in the same manner. You can't every group might calculate it different way. So you have to have access to raw data from all these cohorts.
Yeah.
So that's why I said the raw data is online, downloadable. So you
But
can,
what if these groups, the writers of this paper reached out and that's how they got the data?
yes, so they will do that.
Okay.
Yeah. Some people are not going to share raw data. It's their right.
Right.
But if it's again NIH funded, they have to share. So okay, so this is ITH is bad regardless of histology. But lung cancer, at least this lung cancer, they lumped it all together, non-small cell.
Yeah. No, here they split it.
Oh, squamous and
Here they
okay.
split, they had adeno and then they had squamous.
Oh, okay. So are there a difference?
I mean, in general, no, because at least this is the way we were taught, is because the
Yeah.
confidence interval crosses 1, it's not significant.
Uh-huh.
And is there a trend? Maybe
So-
this one is different.
Yeah, so that one. So that one says ITH. Well, that's really, it's skewed to the left side.
Yeah,
But was that our trend? ITH high was good in squamous, right?
correct.
And ITI was worse in
In
adeno.
adeno.
Maybe this 2016 cohort had more early stage and reflects what we saw.
Yeah. This might be that other paper that I was citing. The pan
Uh-huh.
cancer one. I
Oh.
don't remember it. I just remember Morris.
Okay.
The other one that you were looking at, and you said that this one was newer, so you wanted to. But yeah, so there hasn't been anything definitive. There is another paper that I found that I wanted to show you that actually did something similar to what we're doing here, but let me find it. Give me a minute. Sorry, I just need a moment to find it. I think I may have closed it. Well, anyway, I'll keep looking. I don't want to waste any more time searching, but anyway, so I can look for it while you read this discussion.
Yeah, let me pull that up again.
Yeah, no problem. And then I can look for it in the meantime.
So I guess that paper looked at these two separately, so you can't really say that. I guess prior work has not shown. Well, I guess the clinical impact of ITH has been looked at, but its interaction with TMB or subtype of non-small cell lung cancer has not been
Yeah,
explored or something like that.
yeah. So subtype. No, that makes sense.
So I mean, you say interplay here, but the subtype has been emphasized.
Yeah.
What do you mean there is yet unagreed?
Oh, I just wanted to kind of, maybe my phrasing was bad here, but my goal, the intention here was to say that there really isn't a consensus about how TMB and ITH, I don't want to say interact, but as far as prognosis goes, they're not really used together, and they're not really treated. I mean, they're treated differently. They're just not used together, and it's unclear whether someone with high TMB is always going to have high ITH or vice versa, or if there's some sort of interplay between the two. That kind of idea.
Yeah. It's yet unagreed. I just feel like there may be a better way to say that.
Yeah.
There's yet unagreed upon. Yeah, maybe I can't say it right now, but just think
No, it's
about
okay.
a better, plain way to say, Plain way. So when you say this finding, well, I mean, you said this. Okay. Why did you say this? You think in more genes may be related to resistance? Is that?
What I wanted to talk about here or get at is that tumors with identified drivers may be more aggressive because of those drivers. Those drivers just may lead to a more aggressive kind of phenotype. And as a result, this really goes back to some of the papers I read about the heterogeneity of EGFR and KRAS and other driver mutations specifically. So if there's high heterogeneity, these driver mutations may be able to either avoid detection or able to overcome either immune surveillance mechanisms or T cell cytotoxicity. Basically, I guess what I'm trying to say is, in certain situations like this where there are driver mutations, high ITH may actually help those driver mutations. I don't want to say avoid treatment because there's no treatment here, but it may allow them to progress faster versus situations where there isn't one identified driver mutation or driver gene. ITH, like those tumors don't have the same benefit from it. And I don't know. I'm gonna think about how to phrase this differently.
It's hard to say. And every driver mutation is different. And because this is early stage, EGFR means more a bad outcome here.
Yeah.
But EGFR in stage 4 is best outcome because of the targeted therapy. It's like HER2 breast cancer. Without therapy, HER2 is aggressive. With many good therapy options, HER2 has good prognosis. So here, we're not talking about therapy at all. So we're talking about early stage driver rich or driver addicted lung cancer.
Yeah.
So this is my suggestion.
Mhm.
What we're trying to say here, you wrote, is speculation.
Yeah.
And ITH high just means so many mutations that are subclonal and might affect cancer progression in one way versus the other. Squamous and adeno is different, but we're trying to explain adeno and their higher level of driver mutation. And we said, if the driver is so powerful, you don't need many mutation. But if the fact that you have many mutation may mean that they're even if they're not driver, if you have powerful driver mutation, just like in case of adenocarcinoma with EGFR,
Yeah.
most of them have very low clone number. But if they happen to have high clonal diversity, then what does that mean? In our data, it means high ITH is bad. But is it really bad for ones that have driver versus not? Who drives that worse prognosis? What is your answer?
Well, it seems like based on the mutation frequencies that I had, that I saw, the driver mutations were present at least for example in the high ATH group, so the worst performing group. 60% of the patient samples had an EGFR mutation.
So you're saying high ITH had more mutation?
More driver mutations.
more
There were
driver.
a larger percentage of the high ITH group had a driver mutation than the low ITH group.
Okay. So I think that's the only thing you can say. High ITH in adeno was worse prognosis, but they happen to have higher frequency of known driver mutations.
Okay.
That's it. And then you can say, we know that EGFR mutated lung cancers have higher risk of recurrent disease. And since in the TCGA data, no one received adjuvant osimertinib, which dramatically reduces recurrence risk, what we can say is, we know for a fact that EGFR mutant lung cancer early stage has increased recurrence risk. And if they are and just having driver mutation, known driver mutation in adeno
Mhm.
in ITH group, this might explain the basis of this phenomenon, or it may be
Right.
in part explained. And that's all you need to say. I think what you wrote here is, still so many speculation, and
Right.
viewers will like, why do you, what is
Yeah.
evidence? So I think, I think you can let go of these sentences that I have highlighted,
Yeah.
but focus on what I just said. And
Yeah, that makes sense. That definitely that's clear. That's a lot clearer.
okay. Then the immune chain.
Yeah, and then I just talk about the tumor microenvironment. It seems like in a lot of the more modern, or not modern, the more recent works, this idea of ITH is being consistently paired with TME, tumor microenvironment, and it seems to be very frequently used to explain different prognostic value in ITH, not just in lung, but in other tissues.
Hmm.
And so I just talk about what we saw, how that compares to what's been seen in the other in kind of the literature, as far as these specific cell types go.
Hmm. Yeah, this one's okay. You don't have to use capital for like a member
Oh,
regulatory or macrophage. Yeah. So I would say tumor microenvironment were a little different, adeno and squamous. So here, this sentence seems to say they're similar, but they're a little different. So you put,
right.
emphasize they're enriched, but with different type of cells, mainly, in, squamous, non-squamous there. So just emphasize that.
Okay.
Okay, so I'm okay with this.
Okay. And then I guess what I'll do is the other things I could add to this data wise is the estimate scoring approach that assigns like a stromal score and an immune score. I did some writing about that before. I wasn't sure if that added any value. And then I can also add a Lunit scope.
Okay.
I
Yeah.
don't know if you think there's any value to adding more kind of TME and TME.
Yeah, I think adding Linscope is fine. I think that stromal the score is fine. It's showing
Okay.
difference in immune microenvironment. Yeah.
Okay. I'll add those and then I'll write a conclusion. I was also gonna submit this abstract to JCOPO. Because that's the one we talked about, just to get their input on it.
Huh?
I think that's something that they do, that they offer. They'll
Uh-huh.
look at an abstract and then tell us if they're interested or not.
Sure.
And then that's it.
Very good. Very good.
So I have surgery, my surgery rotation starting next week. So I won't be able to attend these meetings, but what I can do is I can do a little recording
Yeah, send to
of my
Liam.
updates and then send it to, I guess, Liam.
Yeah, and I'll give you input. Yeah.
Cool.
And then you can give Liam like the link.
Yeah.
Yeah.
Yeah,
So,
I'll give them the link to the paper or whatever.
yeah,
I want to
whatever you
show,
want to look at. Yeah.
and then I'll email him the video file.
Mm-hmm. Great.
Can
Good
I
luck
Thank
with
you.
your
just ask you
rotation.
one thing?
Yeah.
Since we're all here, I think previously you said you wanted like about like data from about like 500 patients, but Das told me recently that so far I think we still have to screen out all the patients with their RNA data and then whether they receive the immunotherapy as their first line therapy. But once we filter everything out, we'll have less than 100 patients. And so I'm not sure how we're going to reach the number 500.
Hmm. So Tempest available lung cancer first line immunotherapy, you think it's going to be less than 100?
Yeah, it's gonna be way less than 100. It might be even like 50.
This is only for NMH patients, though.
Mhm.
Hmm. So how many were the radiomics cohort, Chisang?
In total, about 200, but we only have about 100 patients with all clinical data.
All clinical data. And so, Haesung, you think among them, people who have Tempest is going to be maybe half?
So not every patient has the RNA-seq data. Like looking at Stats's list he gave me, he said like 61 patients with adeno, 11 patients with squamous, but not all of them have the RNA-seq data. And so we'll have to filter them out. There's additionally 50 more patients with either adeno or squamous. Those people have RNA-seq data. So once we add them up, it'll be about like 50 or whatever. But then we're not sure about these patients, like lines of therapy, like whether they received immunotherapy as like first or second or third lines of therapy. And so once we filter that out, it would be less than 550.
Nice. Nice.
And previously you said we're going to consider patients who received it as second or third, but it sounds like you just want first line therapy.
Yeah, second and third, I mean, if we're that desperate about more patients, then we can add those second and third and see if we can enrich them. But okay, let's get to immunotherapy. We're gonna do first immunotherapy used, whether it's first, second, or third. Let's do that. Just to catch more patient. But Jisang, when was the last like data point? Like when was the last enrolled patient in radiomics? When did you do you know? Patients' data were collected from year 2000, 19 to 2000, like we're in 2023. So when was the last patient enrolled?
Oh, I don't know exactly, but
This radiomics data has been, but you did add recent new patients too, right?
what means recent patients?
Like you identified more patient. When you first came in, there's radiomics data from Hyunsun.
Mm-hmm.
Did you add more patient?
Oh, so I didn't change the patient pool, so
Okay.
it's only based on
So
Hyunsun's
Hyunsun was
data.
here a long time ago. So, and when we started collecting data, that's probably a couple years from now. So we need to get them all in the Tempest data pool. So I think that would be a lot of patients. Yes, so
Can I just ask Stas quickly a question? So Stas, did you get that patient data from this website, Tempest? And then did you like filter out tests with like RNA like this? And then like diagnosis long? Because once I do this, I only get patients from Dr. Che. Once I do that, like this is the result I get. And these patients are all from Dr. Che. And I don't really see the RNA data here. Like, how did you get that number?
Right. Right. So what I did, I exported all the data and then I did filtering on my end, but I didn't realize I could do filtering on the website. And then what I had to do is I had to query the Tempest S3 bucket, and I would check the bucket to see if the bucket had RNA for every single accession file. So unfortunately, you're totally right. There's no way to tell that patients have RNA data from this. So I had to do it. I had to do it directly by checking the bucket with every single accession number.
What do you mean by bucket?
So, so the way that Tempest gives us access to data is by storing it on a Amazon Web Services server, which is kind of colloquially called bucket, because it's a bucket, I guess they throw all the data into.
Okay.
And it's called S3. S3 is kind of the official name of these buckets. And so I can even share my screen and show you what it looks like, just so that you have a little bit of context for a little bit more context when I talk about it. Where is it? Sorry, give me a second. I have so many things open. Ah, here it is. All right. So this is okay, hold on. Okay, so this is the Amazon S3, which is the way that they set up their servers for Tempest, and the bucket is called TM-Northwestern. And again, when I say bucket is called, what I'm saying is the storage directory, whatever you want to call it, is where all the data is stored. And so here's just lists of accession numbers for all the patients. So if we take one from 2022, TL Tempest Labs -22 is all the patients from 22, and you can randomly click on one, and you'll see that there's CNV, copy number variation, and there's DNA, but there's no RNA. So kind of the
that's
assumption
how
I'm
you
making.
figured it out. I see.
Huh? Go ahead.
Yeah, that's how you determine whether they have the RNA.
Yeah, I mean, I'm making the assumption that if it's not here, they didn't do it. It might be better or more conservative to just say, hey, Tempest, can you give us a list of patients that you've done RNA-seq for? And see if they can do that. That might be easier. And I can ask them as well.
Yes, please. That would be nice.
Yeah, ask.
I just did, I took this approach because I wanted to kind of like get it done in the moment, and I didn't want to wait for them to respond.
Thank you.
Yeah.
Because with what you've told me, we literally have like less than 50, I would say. And I'm just trying to figure out how we can like get more people.
I'm trying to find one with RNA. Oh, here's one with RNA. So there'll be a folder that says RNA, and then we'll have all the files. And then what I wanted to kind of emphasize is when we're talking about processing this data and using it and trying to incorporate it into different things, these files are large. This is 1.2 gigabytes, 2.6 megabytes. The FASTQs, 2.8 gigabytes. You can get them compressed. These are compressed already, actually, this one. So we just need to keep that in mind, because moving this data around is challenging when it's this large. And this is just one patient. So whenever we do work with this data, I think it makes sense for us to have a format, like an RNA input format in mind for whatever algorithm or whatever process we're using, and then cater to that. When we were talking to OncoCyte, for example, they said that we can actually just send them these files, these raw files, and they'll do the processing on their end. So in that case, it won't be that difficult, but that other situations aren't as simple, if that makes sense.
Right. So Tempest, Haesung, you have access to this cloud too, right? This is Stas, but does Liam, do you have your account for this, or?
I do not.
So Stas, they made you this account, but did they make anyone else's?
I'm pretty sure you have login credentials as well.
So is this
they
the
can use yours.
same login presenter for the portal? Is it the same?
I'm not sure. I know that. Let me see. I should have an email from him.
Yeah.
Let me see if I can find it. And I'm pretty sure in that email, you also had credentials.
Yeah, Dr. Chase should have.
So just to save time, just make sure Liam, through my ID, make sure Liam, Haesung, and Jisang has access to the cloud, so they can see the structure of the data, get used to the raw file.
Yeah, it's also kind of a good opportunity to learn about the different output formats, what these data pipelines look like, and I don't know, I think it's just, it requires a little bit of Googling, but it's really
Yeah.
useful to know.
So I'm thinking maybe, um, when you talk to Haesung about the PyClone,
mhm.
or even before, Haesung and Jisang can learn a little bit about the raw RNA sequencing file, the formats and how you extract information, and I'm sure you know about the normalizing, or if it's already normalized or things like that, just like the basics of RNA-seq data.
Yeah,
Maybe you can, I mean, they'll need to learn, but I think
right.
we can maybe spare like 30 minutes to go over with Haesung and Jisang. That would be great. And
Yeah,
feel free to invite Maddie, like, cause she's kind of involved in this project too. So Liam, you can kind of coordinate that, make sure, and you can be part of it too. So how
yeah, I'm
about
happy to,
that?
I'm happy to, I'm happy to do that. I just want to let you know that I'm not kind of an expert on this, so I also have knowledge gaps. So if there's something I don't know, we'll learn together.
Of course,
So,
whatever at your level,
yeah.
that's fine. And the rest of it, they'll fill in the gap. There's,
Thank you, Stas.
Mhm.
yeah, but Haesung, please make that happen with Jisang and Stas at least, okay? All right.
Dr. Che, I know you have login credentials, but I just cannot find them in my email.
Oh, okay.
I know you do, but
Uh-huh. So just send Haesung and Liam the link
oh
to
wait,
the Amazon hub. And Liam just use my own portal credential to log in. And if it can't, I mean, and then I'll look into those emails. If I can't find it, I'll just have to ask, for your credentials, use yours to log in for the time being.
okay, I found it and I'm forwarding you the email, Liam.
Okay, awesome.
Great.
All right, let's do that. Let's do the people who are in Korea first. Juhi, are you able to give us update?
Yep. So I'll just give you updates on like 2 of the things we did,
Mhm.
and then we'll go ahead and give you a full update next week. So I wanted you to have a look at, I'm just gonna share my screen.
Okay.
So if you could have a look at what we wrote out for PSA. One second. This is what the one in blue is what we wrote. If you don't mind skimming through it.
What about sensitivity? Oh, the false positive is the sense, I guess.
They all like from the
What about
data
false-
that they used as evidence, like the only number that we could use was false positive.
Hmm.
The other numbers that they presented was too far, like beyond screening. So they gave numbers for like mortality rates for prostate cancer, how that changes with PSA screening, or like overdiagnosis. Like it was like too far out for us because we don't look at
Those.
anything beyond screening.
Did they mention about false negative rate?
No,
I wonder why.
I think they were more interested in the implication of that for like for like actual cancer, like the outcomes, more than like halting at the screening thing itself. But I thought it was still like okay for us to use it because the false positive that they presented was 17.8, whereas I think our false positive was like super low. It was like a single digit. So we could still make a sentence, but I wasn't
Yeah,
sure what
I'm
else.
okay with this, but I just feel like not just the USPSTF, but also there's gotta be a decent paper citing that we can cite to mention the false negative rate.
False negative.
I
Okay,
know
I
a
could.
lot of tumors that does not shed secret PSA. So that's false negative. You can have very aggressive prostate. And
Okay.
because it's not PSA secreting prostate cancer, you just don't. So, because that's another problem. And compare that to the false negative rate that we have outside USPSTF.
Okay, I could do that.
And then that, a study that led to the 70.8%, you should cite it.
Oh, yeah. I'll do that. It was a European study.
And then that grade C recommendation is coming from USPSTF.
Yeah.
And that recommendation changes over time, but anyway, that has to be cited, like what website, where's that,
Okay.
statement, Yeah.
Yeah, I wanted to show that, and we added 3 journals. So 2 journals for the meta-analysis and
Uh-huh.
all 3 for the actual systematic, the narrative part. So we updated those. But I guess in the interest of time, I just wanna pass it to Eugene so that we can talk about the meta-analysis, because I think that's more important, and it'll be nice to get your feedback on that.
Okay. Okay, Eugene, you're next, but let's take a 5-minute break or 10-minute break. It's been almost 2 hours. All right. All right, Eugene. Are you ready, Eugene? Maybe she's not ready. Tree, on just waiting for her. Do you have anything else to update us?
We rewrote the, if you remember last time I showed you. One second, I'll just show it to you.
Oh, I came back.
Oh,
Oh.
okay.
And yeah. So I have only two things to give you update. First thing is about Meta for MCD study. I'll share my screen. Okay. So new. I think you can see my the shared screen, right?
What is?
Give me a sec. Yeah, I'm working on it. So sorry, I should have pulled up the document, but trying to do that right now quickly. So here it is. By the way, all the presentation for today was, they sound very exciting, and I hope for the best for all of us, all of you guys. And so for this study, so I kind of revised all the part for statistic analysis and also the meta result part. And it would be great if you could just look through the meta part. I mean, only like 4 or 3 paragraphs because it changed a bit. And yeah, that's the
So
main.
what? So explain what changed.
Okay, so like because we included new studies, obviously numbers are changed. But then so I removed all the like I-square thing because they are not recommended by Cochrane. So I replaced the results about heterogeneity, yeah, for meta by just summary RC curve and but nothing that new is up like added. I mean, because summary RC curve is an image, so I added a few like figures, but it's hard to describe that figures, in this paragraph. So that's main change. And also I based on the comment we received from Dr. Hyunjun Shin, I described the like pooled false negative and false positive rates, and that's also utilized for Juhi's like PSA thing. So for us, unlike PSA, our first I think there's a mistake. For our like pooled false positive rate is 0.2, which means like 2%, which is way lower than that of PSA. And our false negative rates is around about like 40%, so like 0.039. And then what I did with these numbers is that I kind of assumed that cancer incidence as like 1%, because that's what I found about that was like an average cancer incidence across like genders around 50s or 60s in the US. So, and an average like age of the population included in our meta is also like 60, so 60s. So, and I kind of try to assume how many extra number of people will be misclassified with MCED tests based on our meta. So I calculated the numbers and I put that in here, and I put the figure as well. And yeah, these are pretty much like change it.
Uh, 100,000 among 100,000. Okay.
Yeah,
That's good. I'm
okay.
yeah.
And then, yeah, other than that, just a quick question is, so I kind of reformat every tables and figures. I made it more succinct compared to the previous version. I put like bunch of forest plots for each like variables before, but I chose to represent them into a nice table like this. And I'm wondering whether you think I need to still have like forest plot at least as a like supplementary figure. The reason why I'm asking this is I have to actually make forest plot again for each like variables listed here, since spec like PLR, NLR, FPR, and I mean I can make them for total, but I'm not sure whether, I mean, we really need forest plot for each like substage and like cancer types. So as of now, we only have this table, and this is sensitivity analysis result based on like study type, gender, assay type, though like results are not like statistically significantly different, which means we, like heterogeneities are not detected, not explained by these factors. And these are like summary RC curve that are newly like added. This is plain one. And I put a few different version based on subgroups. So like B is according, it just shows like whether these variabilities are potentially associated with study types or assay types or like gender of the study population. And
The answer is no, right? Not there's
yeah, yeah, when we did subgroup analysis, it didn't like show that result, but I personally feel these curves might be useful for readers, but I'm not sure. I mean, I can just keep only one, the plain one.
This is the plain one. Okay.
Yeah, not much information about individual studies, but yeah.
I think the case control is this one's good.
You think so? What about, I mean, I personally feel gender shows some trends, like obviously the studies including both genders look more, look having less variability between them. Yeah, but like what I found is when we have only one study, we cannot actually perform subgroup analysis. So sensitivity analysis. So it's tricky. So in this table as well, there's no result for categories having only one study, which means like there's no result for cohort and no result for male.
I see.
Yeah. So
Are
definitely
you,
we need,
do,
yeah.
are you planning to put this figure as the main figure or supplemental?
I think at least we have to at least one SRC carve as a main figure, but because it's the kind of recommended way to evaluate heterogeneity. So
So yeah,
I'm
if
not, yeah.
you, then I would then do a panel A, B, C, D.
Okay, like the way I did it right now.
Yeah, so how many
Oh.
do you have? 4? Or-
Yeah, in total 4.
Yeah, just
Uh,
show everything in A, B, like a panel A,
Okay.
B, C, D.
Yeah. Okay. And then one last thing I want to be confirmed is the FPR and FNR, like false negative rates and false positive rates, like the pooled version.
Yeah.
I originally made this as a table like I did for other parameters, but weirdly I draw like forest plot for this at least. And this is the figure about the paragraph newly added above, like calculating the exact like number of people who will be misclassified when we assume that prevalence of cancer will be like is 1%. I'm not sure whether you prefer, I mean, is it okay for me to put like forest plot only for FPR and FNR, like, or will it be something problematic? Should I like unify the format as a table or a forest plot, or is it okay as of now?
So right now, A and B,
Yes, A and B, like, I'm sorry, like A and B is here. Random effects model for A is false positive, showing like 2%, and B false negative, showing like about 40% across the studies that are included in Meta. And because Phelan for this paper, first positive rate is 0, so it cannot be included in the like calculation. So I delete that, and I explained that in here.
oh, okay. That's okay. So this is, okay. What about the sensitivity and specificity? I mean, I know
I mean-
they're same.
Oh, right. So yeah, it's kind of a different way to express the same thing, but for sensitivity and specificity, I kind of summarize that into this table. I mean, here, this is the core, this is actually the like the pooled value for each different parameters. And it's pretty, I mean, FPR is here as well. It's pretty similar to the ones below, like 0.0, like about 2%. The only reason is actually like these the numbers in this table too, they are coming from the website that I introduced like you guys a few weeks ago. So, and they didn't like provide me with decent forest plot. I have to make that on my own, but then for FPR and FNR, I kind of had to calculate that number by myself. So that's the way I have the forest plot, because I have to make the code for that.
Yeah.
Yeah, so like I'm wondering, should I really make each forest plot for each parameters described in table 2 as well, or is it okay for us to submit as, as of now like this, and then
So if
give
I
them,
remember, last time, didn't you have sensitivity, specificity?
yeah, I kind of, because I at that time I didn't use the website, I kind of used the Markup or a few packages in R, but then one of the packages that I used was actually not recommended by Cochrane, so I actually try to use like the website, which is way more convincing and way more approved by other meta studies. So
So
Yeah.
I think we need for splat for all total.
okay, at
So we
least
have to-
for total.
Yeah, don't do by stage or cancer type, it's too many.
Makes
So
sense.
at
Yeah.
least sensitivity, specificity, so that you can make that like a panel, the A, B, C, D. Still
Okay. And
for the
then
figure,
keep that as a main figure or as a pool.
main figure.
Okay. Yeah, makes sense. Yeah, make that and I'll prepare them for by submission,
Okay.
resubmission. Yeah, okay. That's pretty much
I
all.
I don't know if I like the happy face, this, the red face.
I mean, I cannot change. This is just like format. I mean, if you can give me some what you think might be better, I can at least try to modify this. Or I can just delete the like
Yeah, I think
image.
you can delete
Okay.
the
So like this.
figure. I think,
Or I can make a table. I mean,
so test positive, test disease, and so these are, so I think you can write false positive, false negative,
okay.
all that,
Instead of
because these are kind of a little bit wordy.
mm, okay, makes sense. I'll
Rewrite it, like in PowerPoint, you can use it.
okay. I'll reformat it
Yeah,
more.
test positive, and you can say, disease positive, test negative, and then
Mm,
and then in a different bold letter, false positive, false negative, or all that.
yeah, understood. Okay. Yeah. Other than that, pretty much the same. So like TOO like this and okay. Yeah. Other than that, I have only one thing that you
Mhm.
need your help. So you remember I'm working on MRD as well,
Yeah.
and I received like for swimmer plot made by Natera folks, Natera people, and I'm trying to calculate sensitivity and specificity for that data as well. But I have some concerns whether regarding like classification. So I'll make it very short, my question. So for example, I noticed, first of all, I'm very grateful for receiving this fantastic swimmer plot. So for example, we can, because this is a real world data and this is not like this is real world data. ctDNA surveillance actually, like we can notice that the like time difference or the like the number of times that it is done are pretty like variable among like patients. So I'm interested in calculating sensitivity and specificity between these three like categories that I think of.
Mm-hmm.
So for when we consider ctDNA positive at any time, I
Mhm.
noticed, for example, like patient number 19, we can even like this patient is classified as ctDNA positive because it is converted to positive around this time point. So this is pretty clear to calculate each patient.
Mm-hmm.
But I noticed some papers they only consider like ctDNA positivity at the first time point after curative treatment. So I'm wondering whether it is kind of feasible for us to do or not, because for example, the patients marked with red like mark, I'm not sure whether how I can classify this patient. For example, definitely this patient number 5, this patient's ctDNA is done by technically saying before curative treatment, which is surgery. Would you consider this as like before curative treatment surgery or MRD? Do you think we can consider this as MRD assay? It looks to me like it's done right before surgery,
Mhm.
and obviously we have 2 more assays done after surgery, but like I even try to figure out whether like ctDNA positivity before treatment has any significant like value.
Uh-huh.
So my understanding, the way I approach, I found out 7 patients eligible for this like categories
Yeah,
marked by, I just need you to confirm. Like
yeah.
patient 2, obviously yes. 21, yeah. Like it's overlapping. But to me it looks like assay is definitely done before surgery.
Mhm.
So can I consider them as like this
Yeah,
result
you can.
as, yeah, okay.
Mhm.
Okay. And for surgery, it's easier to like determine, but for radiation therapy, when you look at patient number 25, I will magnify the screen a little bit. Oh, why it's not? Okay. So this patient number 25, I know this patient, she received neoadjuvant chemo, IO, and concurrent radiation as a curative treatment. And her ctDNA was positive even before starting that neoadjuvant chemo.
Mhm.
And so I can consider this patient as ctDNA positive treatment, and also okay, but not um not ctDNA positivity after treatment, right? Because after finishing radiation, it kind of converted to negative,
Mhm, mhm.
luckily. So okay.
Yeah.
Okay. And patient 32 is a little bit confusing too. I think we have to shift the bar a little bit toward right, because it's hard to figure out what was like curative treatment for this patient as of now. So it says this patient received neo as well, and then I mean, this patient looked like having progression even before starting curative treatment. Like red reverse triangle is progression. How would you classify this patient when
I have to look
drawing?
at the history. I don't know what curative therapy this patient got.
Oh, okay. It looks like, I mean, it's a little bit like overlapping. I think this patient also received radiation, but weirdly, before and after radiation, this patient was detected with progression. Unfortunately, and then
Where is the radiation? It's just the color doesn't show up.
yeah, the color. I mean, it's subtle because between like 2 dots of black circle, there is like blue line signifying radiation therapy.
Hmm. So you can, I don't know, I have to look at the chart, but you have to argue that there is. So that looks
So
like concurrent.
yeah, here you noticed, right?
it's a concurrent, that's a curative therapy. So
So,
even after curative concurrent chemoradiation,
uh-huh,
there is,
DNA, yeah, didn't,
Yeah. it didn't
it
clear.
wasn't converted. Okay. So in this case, I can classify this patient, oh,
As pre
sorry.
and post both positive, yeah.
preempt, oh, okay. Sorry. Yeah. And for last question is patient number 33. I'm not sure whether this patient is eligible or not, because it looks to me,
Mhm.
this patient looks to me only having osimertinib. Oh, sorry, this patient got surgery, obviously.
Yeah,
And for this patient, is also like ctDNA positive both before and after surgery, right?
yeah,
Okay. Okay. Yeah. Thanks. I think I can confirm like contingency tables for these three categories and describe the results and discuss.
very good.
Thanks. Thanks for your input. I will try to wrap up the manuscript by our next meeting, with Natera as well. I'm working on it. Trying
Yeah,
to-
this is all written up. They just gave us nice figures. So it's good that they have their own writer who can generate all
Right.
this for you, so.
Yeah, okay. Yeah, these are all the questions that I have as of now.
Do you know where we're submitting it to?
This one,
Yeah,
like MRD,
yeah,
Natera people, they told me that our target is JCOPO, for the first
okay,
target.
that's fine. It's pretty short, so make sure it's meeting their requirements, word limit.
Oh, okay. Oh, okay. Yeah, we'll do that.
Okay, good, good. It's almost ready.
Thanks. Yeah.
Thank you. Um. Anything else, Eugene? Or,
I should have given you like more updates for more projects, but I will do that by next week.
okay,
As of now, yeah.
okay, thank you. We'll do Qisang. Give us an update on.
Sure. Let me share my screen. Can you see my shared screen?
Yeah,
All right. So the first, it's about AACR poster figure for abstract. So I made this. So is it okay with you, Dr. Che?
looks good.
I'll put this figure on poster. And regarding transplantation, I analyzed survival of post-transplantation malignancy and graft failure for each. Oh, it's 6 organ transplantation type: heart, lung, heart, kidney, pancreas, liver, intestine, with UNOS data. So let me show you at first lung data. So the upper left is overall survival according to graft failure, and the middle upper one is post-transplantation malignancy, which is called PTM. And on the right side, it's a graph showing both PTM and graft failure, GF. And the bottom left is a graph of survival analysis based on the number of PTM types. So yeah, and patients without death record was censored. So let me explain one by one. So in lung, no graft failure group and positive PTM group has higher overall survival, as you can see in previous Dr. Jian's data. And in subgroup analysis, the trend is also similar. And with more than one PTM type, it shows better overall survival. So better survivor in 2 and 3 groups. And in heart, it's very similar with lung results. And you can see pancreas. The trend is also similar with lung and heart. So with without graft failure and with PTM, there is better survival observed. And in kidney, the trend appears to be weaker compared to lung and heart and pancreas, even though the number of patients was much higher than other transplant types.
But there is a still statistical significance less than 0.05. So
Right, that's
yeah,
correct.
it shows similar trends.
It's almost
Wait,
yeah,
wait,
it's
wait.
still-
But what's the hazard ratio?
So it's p-value is shown here.
But
And
if
you can
me, PTM longer part is lower.
all right. The trend has changed in kidney. I didn't see that. So with malignancy, there is poor survival outcome, right?
So what is unique about kidney to explain this?
So let me show other organs first. So in liver, it's totally reversed.
Reverse. Wow.
Yeah, in liver transplantation, graft failure affects lower survival, but PTM positive also has low overall survival. So similar in kidney type.
So
And
looking at
interestingly,
liver and kidney, I think we might. So post-transplant malignancy, there's one is contamination or recurrence of the existing tumor versus new type of tumor occurring. So, it's, and then with this data, it's hard to know. So we just have to have a random cutoff. Same organ, and if it was diagnosed within 6 months or 1 year, you can look at the literature. You can say that is a, immediate recurrence of the original tumor. My question is, that's bad, and that if we can separate those two categories, the original organ recurrence versus everything else, you might see a different trend. So maybe that's do subgroup analysis for within malignancy. Does that make sense?
And in liver transplantation, interestingly, in subgroup analysis, you can see on the right graph. So in graft failure positive group, so you can see blue and green one, there is better survival with PTM here.
Oh, interesting.
But in graft failure negative group, the red and orange one, there is poor survival with PTM. So it's, but yeah, anyway, it's obvious, you can see in the graph.
Yeah, so I think, because of the sample size, I do believe this is a real trend. But my question is, are these, let's say, graft failure negative, PTM positive? Are those mostly liver recurrence versus other organs? And are these mostly recurrence within 6 months, 1 year versus, out far? So I think delving into that question and different distribution of that liver versus lung might give us a clue about what's driving this. Okay.
All right. And yeah, small intestine is not solid organ and will not be included, but I put the results. So PTM is not statistically significant with overall survival in intestine transplantation.
Interesting. Okay.
So anyway, overall, the number of the PTM types, the better survival outcome. The trends remain regardless of transplantation type, as you can see, lower left graph. So lung, heart, pancreas, and kidney, and liver.
Did you combine everything?
Not yet.
Okay.
We sorted out by each organs, and when we decide which organs should be included, then we can put together, then make an analysis.
So what are the percentage? If you can, I want to know, what's the lung, among the ones you showed me, is kidney half of the number, or kidneys?
So in lung, about 20,000 patients. Heart, 50,000.
50,000. Heart 50? Lung is 20?
Yeah.
Or heart? Wow.
Right. Heart has more patients.
Yeah, Jisang, actually, when we really studied this project, we have to exclude the recipients less than 18 years old. Because there will be so many infants and child this graph. So we just wanted to look at the trend right now. And then we have to handle the data thoroughly. And then we need to make.
Okay.
I agree. Adult. Okay, anyway, 50,000 for lung, and pancreas is 6,000. Okay.
Oh, 6,000. And kidney is 300,000.
Wow. Kidney.
Yeah,
Kidney top.
that is the main, solid organ in transplant world.
Yeah. What about liver?
Liver is 100,000. About 100,000.
100. Intestine,
Intestine is 2,000.
2000. Okay. Well, I guess liver and kidney, we don't see that obvious trend of PTM positivity. So yeah, I wonder why. And lung and pancreas, and with heart, we do see a trend, right? So I think if we combine it, I think kidney tops everyone else. Even if we combine everything, it doesn't. But go back to kidney. So I guess kidney recurrence versus non-kidney recurrence, we can draw that. We can do within 6 months or within 1 year recurrence versus other and see the difference. Also, we can do within 6 months and within 1 year and kidney recurrence. Put that aside and see if that group and other group shows difference. Let's just try to figure out what explains this. Any thoughts, Dr. Jian?
I have to read the other literatures, but up to now, I couldn't find the exact reference about this study. But there are several studies, mostly it was about the renal transplantation, but they described about the graft failure association between graft failure and post-transplant malignancy. But each article, they didn't show any overall survivor about the graft failure and the post-transplant malignancy independently.
Okay.
So this could be the really first, first study.
Looking at the association together.
Yeah, that means it could be really hard to prove the real research. Yeah.
Yeah.
So definitely you have to work in Korea as well,
Sure.
if possible.
Yeah, of course, it's
This is
possible.
a big project.
Yeah, so it's really interesting.
And you know, kidney is also an interesting organ. So it might have to do with kidney being it's immunogenic, but it's different than lung for sure. It's hypervascular, and its TMB is low, but still it's immunogenic. And this is very different from lung or melanoma. There's something about kidneys, immune system, kidney cancer. So maybe, read about that. This is becoming biologically very interesting. Kidney being hypervascular. In the liver, same thing. There's a lot of sinusoid. Anyway, I can hypothesize that there are higher rate of contamination because a lot of circulating tumor cells will be inside the donated kidney. And you can have higher rate of contamination. And that's not good. And it looks like PTM is like 32.3, so like 10%. That's pretty high for like 1% in general population versus 10% of new malignancy occurring. So I guess the question is, let's say 1% is general population over age 60, like Eugene said. Immune suppression alone, look at the HIV malignancy rate, look at other statistics. It might be 2 to 3%. I don't think HIV people get malignancy like 10%. Maybe look into that. What I'm suggesting is, what percentage is contamination? Or what percentage? So there's donor and recipient. I think contamination from donor can happen, number one. Number two, the recipient might have, undiscovered cancer somewhere, and because of transplant, it just blossomed. So what I'm saying is pre-existing malignancy, not true de novo, occurring, after transplant, whether it's contamination from donor or misdiagnosed recipient. And then there's got to be arbitrary, but a reasonable cutoff to define it. I remember 6 months or 1 year for different papers. So if we can try to separate those and figure out what's driving force behind this figure, that's going to be a good hint for us to come up with any plausible hypothesis to explain this. But again, I think, what is immunological difference between kidney and lung immune microenvironment change? So yeah, I mean, more questions than answers, but I think just describing this begs for a nice paper. But I think just a lot of subgroup analysis to answer many different questions. And also, I said graft failure, for each organ, what is the, rejection percentage for all that, should be looked at. So I don't think we have to do pooled analysis. I think kidney is going to drive everything. But so what are other organs that you haven't analyzed? Is this everything?
Yeah, everything. 6
Oh.
organs.
Okay. I think because this is all the data that we have, we can pull this data together as it is. It's okay to add intestine.
Yeah. But how about the intestine? Because most of the literature says about the solid organ transplantation, aka SOT. They talk about liver, kidney, and lung, pancreas, but not about the intestine because it's not solid organ. I'm not sure the immunosuppressant management is different between these two organ types, but I think rather we exclude the intestine.
I mean, it's very small number, and if you think prior literature only looked at solid organ, I'm fine with, because intestine didn't show much, right?
No, there wasn't significance.
Yeah, and it's not going to affect the overall data either. So yeah, I'm fine with excluding intestine. Okay. Anything else, Jisan?
Yeah, In terms of patients with any previous malignancy before the transplantation were, yeah, they were excluded. I just want to add that. All right, so yeah, and I developed machine learning model to predict the occurrence of graft failure based on 180 parameters with values in as many patients as possible. Also using UNOS data. So I can show the result. So about importance, factors with high importance have an important role in determining the performance of the machine learning model. That means factors with high importance can be used to train a model.
Hmm.
So that means, so you can see
What is
here,
LOS?
LOS is length of hospital stay,
Oh.
and age is age, PO2 is O2 saturation, hemo.
And is this just
I'm not
this is just lung?
It's, yeah, in lung transplantation.
Oh, okay.
Yeah, anyway, the predictive model also lack of accuracy. So you can see this matrix, the real value in row and the prediction is in column. As you can see sensitivity, specificity, PPV, and NPV.
Hmm.
So it's not still,
But why
yeah,
sensitivity specificity is pretty good?
because it's skewed because of the sample number.
Hmm.
So the with how can I, so there is much more number of patients without graft failure, I think so.
So what is the true graft failure number? Is that, what is it?
So this one. So
9?
right. True positive.
But what is it? So where is the assay model? Where is the real? Okay. What's the reality? Which one? Y-axis?
I don't understand what you're saying.
So disease and assay. What? So disease is
So oh, let me check.
usually. So graft failure, is it 200 or 30? How many graft failures do you have in lung cancer? Then we should know the answer. The raw number.
This is not about the patient who had lung cancer, but received the double lung transplantation, right?
Right,
Or
right.
just
It's
lung.
not, yeah, just
Yeah, in total
well,
number is 23,000, no, it's almost 24,000.
Hmm.
And there were 2,316 cases of graft failure. And percentage was 9.7%.
So why there's 20,000, but why do you have 2,000 here, 200? I don't understand the number.
so it's, so I
Oh, it's
partitioned
just-
two
like
sets.
two different cohorts? Did you?
Yeah. in
Oh, I
tot-
got it. Same cohort, but I used the graft failure from the cause of death. And Jisung used the real graft failure data.
Oh,
right?
So I used whole lung transplantation data to analyze this, and there were about 10,000 patients. I separated two sets for validation and a test and training set.
uh-huh.
So it shows the test set results based on the machine learning model developed by training sets.
This is a validation cohort,
Yes, that's correct.
but validation this is a, let's say, like, 250, well, 2,500, let's say, patients. Which is much less than 25,000 total. So you're validating in 10% of randomly picked patients, I guess. And your graft failure rate is about 10%. Is that true?
So so the real number of the graft failure positive is 264, and without the number of without graft failure is 2277.
I see. Okay.
Number one is a positive.
Let me check again.
So just for us to understand,
Easy
I don't know about Yuri's algorithm, but mostly if you have 25,000, you randomly divide them into test set and validation set.
Right.
But here, sometimes higher, let's say 2/3 training set, 1/3 validation set, something like that. Here, do you know what percentage you did training and what percentage you did validation?
So it's for training set 70%, test set 30%.
Oh, okay. And do they, like a bootstrap, do they do it in different randomly pick 70 and 30 different ways, or is it like a one-time thing?
It's about 100 times
100.
randomly selected. Yeah.
Okay.
Let me check. Graft failure.
Yeah, I mean, the specificity is pretty good, but sensitivity is terrible. So it's not a good model.
Mhm. They can't predict.
Yeah.
Yeah, one is positive, two is negative. Yeah, that's correct.
Okay. Okay. Is this all you did? Okay.
Yeah. So yeah, I will sign out the any transplantation types, the organized files from the beginning, and I will also share the status data code for utilizing by other lab members, and I also signed out RadioMix project, collected clinical data and data code, and OneDrive shared by Yuri. And actually, it's not done yet with collecting scan data and tumor information for image trial. So I don't think I can finish until the end of this month. So I also sign out this to Dr. Jian. And also I have a plan to teach everything I learned. So how to sort and organize data using Stata guide. And also I'll teach about what I learned about our code from Yuri. So it's about how to develop machine learning model to predict outcome with nominal or ordinal values. For example, yes or no, or 0, 1, 2, 3, like that. So I will have teaching session tomorrow. Yeah, that's all.
Very good, very good. I'm sure you learned a lot. Yeah.
Yeah,
Yeah.
definitely.
Yeah. I mean, I guess we learned the value of accurate data. We learned the value of comprehensive data, and then how you analyze it makes difference. I think, I mean, applying the AI model to different layers, modality, essays is important. So yeah, thank you for the all your contribution on different projects. So good job. So next week will be your last lab meeting. Is that
Right.
Yeah.
Right, in my last day.
okay? All right, very good. Jian, let's go to you.
Okay, I'll share my screen. Now the general site is trying to ask both decision in progress. Yeah, I'm still waiting. Well, yeah, I hope it could be a revision, but I'm not sure if it is rejected. I'm not be hurt, and then I will just immediately submit to other journal.
Uh-huh.
And now these days I'm working with Andrew mostly about the comprehensive landscape of de novo malignancy after double lung transplantation. So first of all, I want to make sure, do you prefer to use a de novo malignancy after double lung transplantation or post-transplant malignancy? Because I need to decide the term to write the article. There is no official term in other references. They call it sometimes de novo malignancy, sometimes a post-transplant cancer, PTC, or sometimes PTM.
What's more widely used? You know, it's really just to help readers.
There was only one article used post-transplant malignancy, and there was only one article used post-transplant cancer. But most of the other articles used de novo malignancy.
I think de novo PTM usually used in table 1. I think is good.
De novo PTM.
Yeah.
De novo PTM. Right. And can I explain?
Yeah, right here without PTM, with PTM, I think they would understand. You don't have to write de novo here.
Okay.
because it's in the,
Okay.
yeah.
Then you want to use this word?
So when we say de novo, can we differentiate, what is recurrent, what is the new primary? I feel like there's that notion.
We can distinguish. We excluded the other recurrence.
Uh-huh,
So it could be real de novo.
real de novo.
And yeah, as far as I know, in the transplant world, the most of physicians use de novo. So
Hmm.
that's why Professor Anki is supposed to use this term.
Hmm. So de novo malignancy after. Yeah, I think we could keep using de novo in our text
Mhm.
script. Yeah.
And in table, still we can use PTM.
Yeah.
Is it okay with you? All right. Interestingly, in male recipient, they had more PTM.
Oh.
And well, smoking history, because there were many unknown histories, so I can't rely on. And here you can see death. Final events, final percentage is larger in with the PTM, but interestingly, until 10 years, overall survival was higher with the PTM group.
Mm-hmm.
But after 10 years, which were 15 years and 20 years of overall survival, was higher in without PTM, because the cancer is a long-term complication of transplantation. So in later period, there are more deaths with the PTM group.
Oh. I mean, that's the cancer, right? That's,
Yes.
huh. Okay.
This is the incidence of multiple cancer.
Mm-hmm.
This is the number of post-transplant malignancy. It's 1, 2, 3, 4. Yeah. Of course, the skin cancer was the most common cancer.
Oh, because it's most common, liver and kidney transplant data, we could kind of see skin
Yeah.
cancer.
Yeah,
versus
we're
lung
gonna see. Yeah.
cancer. Yeah.
Yeah. But skin cancer is the most common malignancy in all types of transplantation. So yeah, I would say. And this is a supplementary table, but the indication for the lung transplantation, the most common indication was idiopathic pulmonary fibrosis, usually interstitial pneumonitis. And second one was the COPD and emphysema. Third one was the cystic fibrosis. And this table should be the supplementary. This is not important in this article.
Okay.
This is cause of death. Well, Andrew will show you the figures, but I had to decide whether the expression of a cause of death, do we need to distinguish by each causes or by year? But I think the by year would be more important, and it could be more easier to to describe what we found. So yeah, you can see the figure. It would be more easier. Okay, Andrew, are you ready?
I'm ready.
Yeah, this is all I have about table. Yeah, share your screen.
So we got 8 figures today. Figure number 1, total incidence of de novo malignancy. This is it. So after listening all your feedbacks, I've changed everything you wanted to change. Are there anything else you want to say?
Looks okay. Y-axis, you have to put number of patient, number of cases or.
Ah, yeah.
Do you want to use the term cases or just blank and
N,
number?
I think
Cases?
these are patient number, right? Number of
Yes.
patients. Yeah. Number of patients is fine.
Yes, sir. Figure number 2, annual risk of de novo malignancy after
double lung transplantation.
Yeah.
We prepared a barge.
There's an echo.
지하 마이크 좀.
Sorry.
So we prepared a bar graph and pie chart. So this is a bar graph. Bar graph option number 1, we have legends on the right side here. And we got option number 2, we got legends on the bottom. Which one do you prefer?
Okay. So this one versus down.
This one versus this one.
Yeah, the bottom looks better to me. Yeah, so looks good. This is better than the previous one you showed me. Yeah.
Yes.
And
This one is a
So
pie chart.
always bold all the letters and numbers. Make it bold. When it goes, it's hard to read.
Because the letters were so small, when we make it bold, it could be so thick.
Can you
Okay.
try it here? Yeah, I think it reads better. Okay.
And are you
This
okay
is-
with the color?
Yeah, I'm okay with the color. So for prostate cancer, why does only have grade? Oh, is it gradation or what is it?
All different types.
Oh,
If you want to, we can change the color.
yeah, it feels to me like a one gradation, one color to me. So
Okay.
make it like all different colors
All right.
so that you could see there's many different types within that gradation. And also put the cancer in the order. Is it in the order that was described?
The orders were decided by total number.
Okay. Total number. Okay,
Next one, pie chart.
This was the one you very firstly ordered.
Yeah. I know they're sending same message, right?
Yes, we should decide between slide 7 and slide 8.
Yeah. I think 7 has more information, like the absolute numbers there, so cool. Yeah.
This one is figure 3, cumulative risk for top 4 cancers.
Looks good.
This one is figure 4. So this is the incident rate pie chart. By age.
So this is what I
wanted to ask. We can put the legend here, or we can put these big legends as like this. Which one do you prefer?
The right one. So skin cancer, you could use basal cell skin cancer, squamous cell skin cancer. You don't have to put parentheses. So Andrew, why don't you just write it down? We're recording all this, so you don't have to use your time to write this. Go to the next slide.
This is incidence rate by age.
So a lot of skin cancers in older. Lymphoma is younger population. More, I guess, more lung cancer in elderly, which is kind of the general population's trend. I think the legend can go on at the bottom.
Okay.
Like you did for the, yeah. Next, you done?
This is overall survivor. As you can see, the right side one is de novo malignancy after double lung transplantation. And the right side one is according to the numbers of post-transplant malignancy. When you look at the right side, There is no enough space to write it down the legend on the graph.
Right.
Yeah, it is overlapping, but I put the skin cancer on beside of the blue graph.
I see. So I think just like the one in the number of malignancy, you have 1, 2, 3. I think you just need to get that on the right side and just put the legend on the right upper side of the first graph. And by color, you just put the legend.
Okay, right upper side, okay.
Yeah.
And the right one, do I need to use as English as ONE 1, 2, 3, or just real numbers?
Number 1, 2, 3 is fine. But 3 is equal and greater, right? So here.
Oh, yeah, okay, yeah, sorry.
So just an insight coming from this graph is that lung cancer is worse. So this is a double lung transplant data. So if you have lung cancer recurrence, whether it's true, lung cancer recurrence of the prior lung cancer, or if, this doesn't distinguish de novo happening like 3 years later, whatever that is, if lung cancer is the PTM, not good. It does not. So, I'm wondering, for kidney cancer, if there's a lot of kidney cancer recurrence, the graph might not be as good as, maybe it's gonna follow this trend. So, kidney and liver, you can draw a graph like this and see what drives worse prognosis for those.
Andrew, can you go to supplementary figure about the overall survivor? Yeah, slide 20. There are more worse cancers than lung cancer.
Lung cancer is the blue. Yeah. Light blue, right?
Yes.
So many. What is the purple that's worse?
Okay, give me one.
The purple one that is below, yeah, the lung cancer, that one. That looks pretty bad. There's so many different purples. So it's hard to know.
Because these are 27 types.
Yeah.
It doesn't look so good. So I'm thinking about what if we cut 10, 10, 7, or because we already showed the top 5, then 22 left. So we can make 2 other graphs with 11 each malignancies.
So we did top 4, right?
I think it was top 5.
Top 5, okay. Yeah, this is too much. So you're suggesting 11 from how did you select 11?
In orders.
After top 5?
Yeah,
Okay. But why 11?
oh no, I mean, because in total of 27 cases, we used already top 5, then the 22 malignancies left. So I can divide as a 2 different graphs with each 11 types,
Oh, I see.
or 3 graphs.
Yeah, let's do if we will. There's 22. You can do four graphs A, B, C, D, and just show that how other, yeah, let's do that. Four graphs and make a panel A, B, C, D, yeah. In the order of frequency
Okay.
after top 5, yeah.
Andrew, you can keep doing.
So this one's a cause of death by year, or with or without double lung transplantation malignancy.
So again, same thing, the last few ones, just make it very distinct, different colors. Because it's hard to, yeah, identify them. So, okay, show me the next graph.
When the recipient had de novo malignancy on the 3rd year, there are many deaths occurring.
And that death is malignancy. A lot of malignancy, but then also a lot of graft failure.
Yes.
Wow.
So I wanted to show by years.
Yeah, so this is with malignancy. Show me the other. Oh. So here, this is a very interesting finding that with malignancy, there's a lot of cancer-related death. So I guess cancer being the cause of death peaks at 3 year. Here, okay. So when you go to the next one, yeah, so this de novo malignancy causing graft failure with chronic transplant rejection. So to me, this graph, so like malignancy and graft failure go hand in hand, at least in 3rd year. Yeah. Huh. Yeah, so this is really, why is 3rd year this is cause of death and malignancy. So if they develop cancer, you know, they struggle with that cancer for like a year or two and they die. So. Oh. Okay, this is okay. I mean, like just to present, but I feel like, I don't know, but maybe that lead time bias where survival bias plays a role here. Because, so how do we generate this data? De novo malignancy. Let's say a person develops first cancer at year 4. That person belongs to de novo malignancy group.
If it is the first occur,
Yeah. Okay. Okay, for now, this is okay. This graph is fine. The other one, AB, I'm fine with it. Yeah. So for infection, COVID and bacterial, I think can be combined. Because right now COVID is not the, whether it's the infection or not is the only important.
oh, viral plus the bacteria together.
But then this one is COVID specific. Yeah. So you could just combine it in pneumonia as infection. Yeah.
Okay.
Okay. Anything else?
Can you go to supplementary figure? The next one. Yeah, slide 22. These are the same, but it is just we divided by cause of death, not by year.
Oh,
First, we made this figure, but then we realized that on third year there were so many deaths occurred in post-transmalignant group, so we made it again by year, and we've made it the previous one.
I like this too. Let's use it for supplemental.
Thank you, Andrew.
I'd like to suggest just
Yeah.
one thing. So could you show us figure 2? Is it cumulative incidence graph? Oh no, it's figure the
One.
more upper figure. Maybe next one, figure 3. Right here. So I'd like to add the colorectal cancer in this figure because I saw in figure 2, colorectal cancer is quite late occurring cancer in
Yeah,
the figure
actually,
2.
first we made the top 5, but the problem was the colorectal cancer and lung cancer, they were overlapped. The very similar graph they shown. So that's why we removed the colorectal to show the lung cancer trend.
Oh, I see. Okay.
Okay.
That's awesome.
Yeah, Andrew, I think we're done with the figures. Can you unshare your screen?
Okay.
And the next project was also presented by Jason. So I think we can see the overall survivor totally and with each organ. And we can combine that and make analyze the association between two vectors. And as you supposed about the bias, we're gonna see. And well, there are the diagnoses of date, so we can figure out the first occurrence, the second occurrence, and third occurrence. And maybe we can ask to Dr. Shin about that phenomenon.
Yeah, so do you agree, Jian, that it can be a bias?
Yeah, it could be. First of all, we have to see the data, but if there is some kind of a bias, then we have to describe about the reasons or the other theory. So then we can ask to Dr. Shin.
Yeah, so remember we talked about landmark analysis. That could be one way to overcome it. Because let's say a person develops cancer at year 3, 4, 5. So you can do landmark analysis at year 3. Anyone who makes it to that point.
Yeah.
Right? If they died of graft failure year 1, I mean, you know, then you're gonna, if you include all of those, then who survived till year 3 has chance to develop lung cancer. Or like,
Yes.
if you, and that's going to say, you know, if you develop cancer, you live longer, but that's really not the case.
Yeah.
Because most people died of graft failure before. So if you do landmark analysis at different time points, year 3, year 5, year 1, and only analyze people who made until that time point, and then do similar analysis and see if it really holds true, is a simple way to overcome that bias. And if we do that and say, well, the graph looks very similar, and then we can still say that the bias have been looked into. And yeah,
I understood.
Yeah.
Okay. The next one, we're still waiting the next result. Yeah. That's all I have today. Thank you.
Very good. Oh, Jian, did you look into the PARP inhibitor question or?
Well, actually, I couldn't find the accurate reference what you asked me. I'm still looking for, but until now, I
want to make sure you understand the question. So can you
yes.
repeat the question you're trying to answer?
There was an event about the regarding the ovarian cancer.
Yep.
They used a PARP inhibitor, but and until now they used it, but now they stopped using because of their fatal phenomenon after using that PARP inhibitor. And then the other solid cancer regarding breast cancer, pancreatic cancer, or prostatic cancer. Is there a similar phenomenon in those cancers? That's the first question. And second question, then what was the mechanism about
Right.
this?
Right. Yeah, at least in ovarian cancer, the companies are not developing PARP inhibitor for that specific indication. So what was that trial that leads to PARP inhibitor being withdrawn?
Mhm.
Withdrawn. Yeah. And what, what's that instant? So there is definitely a caution of using PARP inhibitor plus the immunotherapy as a combination. There's some people not wanting to do that because of that negative. So the PARP data has not panned out to show, it showed worse survival. And what was that population and how do you explain that data is the question. So keep reading
Okay.
about it and let us know what you think.
All right.
Okay. Haesung, do you have update?
Yeah, I do. I want to first show you the DART trials for desmoid and Volver.
Yeah.
Here's the introduction I wrote for desmoid. Oh wait, am I sharing the screen? I'm not, right? Yeah, this one.
So this is your desmoid introduction.
Yeah.
Okay. Okay. So looks good overall, but I'll go with you individually once you print it
Oh.
out to me because for the sake of
Oh, okay. Yeah, sure. Okay, then, and then also had a question about the grant that you sent me. Should we talk individually about that as well?
individually, yeah.
Okay, and then the Tempest tap, last time we talked and we had a bunch of questions. We decided to talk tomorrow in the morning, so that's that. And then,
Okay.
yeah, I guess besides that, we already talked about transcriptomics. I have journal clubs. I have 3 topics, but we can set that aside for another time too.
Yeah, just because sake of time.
Yeah, sure. Okay, that's it.
Yeah, thank you. Myung-woo, do you have update?
Yes, so I wanted to show you some PowerPoint slides. So I ran some prelim analysis and graphs, not the full one. And my goal is to complete the figures tables by next week, at the end of next week, at the latest. So
Okay.
based on our discussion today, I just wanted to ask what kind of graphs you want, any additional analysis that you want me to run, and
Okay.
some visualization of the data. Let's see. Let me see my screen. Yes. So this is a study population. There was 496 in TCGA, but I trimmed some data that didn't have mRNA, XL, or LUNAT data. So I used 469 as a main data, and I used the other kind of data sets for analysis involving mutations or neoantigen or both.
Okay.
So these are my variables, independent variables: mutation profile, mutation count, TMB, neoantigen, thyroid differentiation score, and immune phenotypes. Dependent are the cytolytic score, XL infiltration, immune modulating cytokines, PFS, OS, and for some variables, GSEA. This is a future plan. So this is the visualization for mutation profile. This was the I analyzed driver mutations versus no driver mutations. I
Okay.
included 4: BRAF, RAS, RET, ANTRAC, and no driver.
Yep.
And they're kind of mutually exclusive. You can see here, only one overlap between KRAS and BRAF. These are the percentage, 47% with BRAF and the rest. RAS was the second most prominent. I just added TERT at the sidetrack because I found a paper saying TERT promoter mutation tends to co-express with the BRAF mutation, and when they're combined, they have poor overall survival.
Mm.
So, and also I looked into genes regarding to MMR and HR.
Uh-huh.
There were like 24, but I used the OncoKB. This is like what I call the database from the MSKCC,
Yeah.
and only 5 were oncogenic. So I didn't run the statistics for this. I didn't feel like the need for that. So I was wondering if this should be fine if I just mention it in the paper. Say like 24 were discovered, but only 5 mutations were like oncogenic and 19 were of unknown oncogenic origin. Or do you think?
Yeah, just describing is fine.
In regards to mutation count, these like not many mutation counts. So
Mhm.
it's from 1 to 5. Neoantigen 0 to like 1 is most prevalent. There were some Spearman correlation, which of course makes sense. Not much information regarding this. The median 6 for mutation count for neoantigen, median is 1 and nothing much.
Mhm.
TDS, same thing. So this is inferred using, I think, like 16 thyroid kind of function genes, mRNA expression. Median was like this, and the how they are distributed. So yeah, so the positive means that it's more differentiated, and the negative means undifferentiated. Immune phenotypes, 48% was immune desert, which means there's like no lymphocytes in both the cancer stroma and the epithelium. Excluded is lymphocytes in the stroma, but not in the epithelium. Inflamed is like a lot of them. So yeah, so 0.82%. Prelim analysis regarding mutation profiles. So cytolytic score BRAF was higher than non-BRAF wild type samples, which have been described in the previous paper also. And I looked into other things. Actually, this fusion genes, RET fusion, INTRAC genes, when they have fusions of those, the cytolytic score was higher, and the RAS was like the lowest, and this BRAF and no driver was like in the middle. To see this comparison test, yeah. And so I just looked into BRAF TERT because there was something about the OS, but there wasn't really a difference in the cytolytic score. Mutation count didn't have any significant difference. Neoantigen also no significant difference. This is like a heat map for the immune cytokines. So I was wondering, do you think I should add the heat maps or just like these kind of things, the box whisker? Because I don't think this like gives like too much information or like have like a benefit in visualization of the data.
So what does this tell you? Driver versus no driver?
So I plan to do my plan was to do BRAF mutation, RAS mutation, fusion, and no driver. But while I was doing it, I left out the fusion genes, which is like 10 to 20 samples. So this is actually up to here, IL-12. This is the immune inflammatory cytokines, and these are the what do you call forgot the name.
And this is the mRNA expression, right?
This is the mRNA expression.
And let's say group A and B colors are different.
So if you see here, so mRNA expression, this green side is like a negative, so it's like less expressed. If you see RAS, both the suppressive and the inflammatory like cytokines are less expressed compared to other groups.
So did you apply this to two group?
This one, I think I ran the Kruskal-Wallis, and they were all, I think, significant, statistically significant. So what I was thinking, RAS had less cytolytic activity score and like less immunogenic. So I think this figure shows that the less immunogenic effect is not really dependent on the immunosuppressive signals like CTLA or PD-CD1, PD1 or PDL1. Actually, I didn't really dig deep into the analysis, like how to explain this. I just wanted to double check like what kind of graphs is better for the paper.
Yeah, so my question is, show me the next graph. I want to see what
Yep.
you. So this is like an individual gene expression.
Individual, yes, gene expression.
So not related to heat map. So
No.
you need heat map when you're grouping,
Mhm.
and then you're gonna see that group A and B have different survival, different prognostic indication. But you haven't done so, right?
The survival, I have this here actually. There was no significant difference between groups.
Yeah. So, I mean, because this one you have Lunit score. So if you can do like Lunit score 3 different types, and within those types, see if the immune signatures are differently aligned. Like, or you can do heat map and then a color immune exclude, all differently.
Yeah, so actually I kind of have that here. This is based on the immune phenotype.
Yeah.
And I have the description of how the mutation is distributed in the visual phenotypes.
Yeah. So the other one, we're not using it, so we don't need it. But this one, the immune unit score, yeah, you should use that. As a
So
main.
I was wondering, should I describe all the like landscape or just focus more on the Lunit?
Focus more on the limit. This one, you don't need it because we're not using it.
Okay. So this one doesn't need it. So I want
That one can be all supplemental. This one, this can be all supplemental.
Okay. Do you think I should increase this as well as like a?
Because this is not related to.
Not Lunit, just
But I think supplemental is okay. People probably want to know, so.
Because and this PFS OS is actually interesting. This just happened like 40 minutes ago. So I ran between BRAF plus TERT samples compared to BRAF only samples. And PFS didn't show any difference, but OS had like significant difference actually. So when
BRAF hurt.
So when they co-express, it's like very worse prognosis. And this is actually kind of rare in thyroid, other statistics.
So is there a biological rationale that the
I don't think so, because the other paper that looked into this mentioned like, not sure actually. They mentioned BRAF and TERT promoter mutation occurs like simultaneously a lot. But I'll have to read up more about it.
If it's biologically makes sense that BRAF is influenced by TERT. If there's a biological rationale, then maybe fine to show, but let's revisit that. You should read more about it.
I think it might be just sporadic 2 events like happening together.
Okay.
So this is just TDS with the cytolytic score very low, like R. Same thing, mutation count, So I just use like Excel Prism R, like just to like make a quick graphs because I didn't have much time. But for the actual graphs, I'll have to use more like make a better graph. So this is like TDS with cytokines. So based on the TDS, every cytokines, the inflammatory, suppressive cytokines, both showed good correlation. So the more well differentiated the samples are, the less expression of those cytokines, both of them. And so I'm not sure. So should I like try to describe TDS with the other variables as well, or just not like BRAF kind of things?
I think you can show all you can put all of those
Mm.
in supplemental. It's fine.
Okay.
The ones that has Lunit score is in the main figure.
Okay. So Lunit. Okay. So this is the ones related to Lunit
Yeah.
phenotypes. So in regards to BRAF mutation, this is just the absolute number of BRAF mutated sample versus wild type based on the phenotypes. So if you see the right one, the percentage, excluded has like 83% with BRAF mutation, inflamed 62, desert 40. So when I did a chi-square, was significant, and the postdoc analysis also showed that all three pairs were different in BRAF, what do you call BRAF expression? I mean mutation. So yeah, so I kind of characterized the excluded sample as BRAF mutated samples. It's the same thing.
I think you can in the discussion say a lot of lung cancer EGFR mutated. BRAF is the main driver in thyroid, and EGFR is a main driver in lung cancer, and they are most likely all deserts.
Desert excluded.
Excluded. Well, that's a good question. You have long data. See if EGFR mutation follows the same trend in lung cancer.
Oh, you mean BRAF or?
So EGFR in lung cancer, BRAF in thyroid.
Oh, you mean the main driver?
They show the same pattern.
Okay.
That's a good question.
Okay. Yeah.
Okay.
And in regards to non-BRAF driver, oh, here. So excluded, of course, due to BRAF, prevalent BRAF, they only had 6% without any driver. Inflamed and desert had 29 and 22%. And for the specifics, RAS mutation, desert was characterized by RAS actually.
Oh.
Yes. So if you see the total RAS is like 50 in the whole sample, and 46 of them was in desert. So desert was, so I thought like we can characterize even desert samples as RAS mutation and some of non-driver samples, which makes sense.
Yeah, it makes sense.
Yeah.
So this is actually a good theme. And melanoma is a great, BRAF, right? And melanoma has a lot of RAS.
Yes.
So if you can show same phenomena in melanoma, and melanoma, there's a lot of inflamed, but still some are desert. And if you can show that RAS mutation is concentrated in desert, like relatively, in melanoma, lung, and thyroid, at least start there. And we have, of course, pancreas, but pancreas 95% have KRAS mutation. So it's hard to show, but that's why pancreas is mostly immune desert. So
I'm sorry.
yeah, so I think you could kind of plan for like a pan tumor. Is RAS being a driver mutation, like it's tightly related to a higher percentage of immune desert.
So there was this short paper describing KRAS in lung NSCLC, saying that KRAS is involved in immune evasion.
Right.
And I think Looney paper, Dr. Chong-Han Park paper actually mentioned similar things.
The KRAS was more desert.
KRAS pathway, the enriched KRAS pathway is like regarding to that. But I think in that paper, KRAS was actually shown in inflamed, I think, if I'm not mistaken. So it's like a little different.
Yeah, so yeah, let's see.
Yeah.
But yeah, yeah, remember that. Yeah.
So
Next.
yeah, so this is the one that I mentioned before, the same thing. So this is why I think GSA will be needed, because inflamed doesn't have like distinct signals.
Right. Yeah.
Maybe it's only discernible by TILs. So we're not sure. We have to look into this and see.
Right. Right. Okay, guys, I have to leave for the next meeting, so I'll end here and maybe we can resume later. And then, yeah, the questions you can bring to me individually. I'll talk about it in the afternoon.
Yep. Thank
Okay, thank you.
Thank you.
Thank you.